Informing early, informing better. The Target Translation Consortium of Singapore presents Hitting the Target: How Preclinical Target Validation Can Increase Your Chances of Success in Drug Discovery. Where potential drug targets were once hard to come by, the Human Genome Project has made the genetic content of all human DNA publicly available, opening a new era for therapeutics discovery and development. Of the estimated 30,000 genes in the human genome, only a subset express proteins that are able to bind drug-like molecules. In other words, that are druggable. Of these, a small fraction have known functions that are linked to disease. Hence, researchers today are faced with a different kind of challenge. Much like sifting through a haystack, an important task in drug discovery is now to identify and verify potential drug targets that are instrumental in causing human disease. This is the process of target validation. Validation is a crucial step in drug discovery. The only way to be completely sure that modulating a target gives a desired therapeutic effect is to test the idea in human clinical trials. However, clinical trials clearly cannot be used in early drug discovery. This is why a potential drug target must undergo a preclinical validation process, utilizing in vitro assays and preclinical disease models to systematically confirm the target's role in disease before entering a full-fledged discovery program. Once a target enters a company's pipeline, it can take 10 to 15 years to bring a drug to market, with total R&D costs coming close to $1 billion. Drug development is a huge commitment, and investing early in rigorous preclinical target validation will increase the chances of success. Unfortunately, failure in clinical trials is a major problem facing drug development. Over 95% of the time, drugs tested in oncology clinical trials do not advance to regulatory approval. Among all experimental drugs tested, less than 10% make it to market, and most drugs entering phase 1 and 2 clinical trials eventually fail. Why do drugs fail? Lack of efficacy is a major reason, accounting for more than half of all clinical failure. Another important reason is safety or toxicity. Often, these reasons for failure arise due to inadequate preclinical target validation. Selecting the right target is one of the most important decisions in a drug discovery program. A safe and potent drug, for example, will still fail if a team is working on the wrong target for the disease of interest. Hence, an important goal of target validation is to establish a clear link between the target and the disease. In this retrospective analysis, AstraZeneca found that 73% of projects with genetic data linking the target to the disease were successful in phase 2. In contrast, more than half of projects that failed due to efficacy did not have genetic validation. In addition to genetic evidence, developing a good platform of preclinical evidence using in vitro and in vivo disease models can also increase the confidence of translatability to human disease. Shown on the right, the availability of biomarkers was another important factor for success. 82% of projects with biomarkers available from the start were successful in phase 2. Predictive and pharmacodynamic biomarkers are important to help guide patient selection and to provide insights about clinical proof of mechanism and proof of concept. Importantly, projects with demonstrated proof of mechanism have better outcomes and are more likely to progress to Phase 3 and launch. By building a framework for establishing a strong target rationale, you will be earlier and better informed and have greater confidence that modulating your proposed target will result in a desired therapeutic effect. 40% of the world's pharmaceutical resources are dedicated to cancer, but only 3 to 5% of oncology drugs advance to receive FDA approval. Despite the huge investment to find new targeted cancer therapies, the number of unique oncology targets that are paired with marketed drugs remains relatively small. As a consequence, we find ourselves in a scenario today where pharmaceutical companies are developing drugs against the same known targets. Research from Cold Spring Harbor Laboratory may provide a clue on where or how drug makers can find effective solutions against cancer. In a cautionary tale, this study reveals that drugs aimed at hitting certain targets in cancer may not actually work as initially designed. Target misidentification could be a reason some cancer drugs fail in the clinic. Using a CRISPR-based knockout system, researchers investigated a set of cancer drugs and their putative targets at various stages of clinical testing. They found that all the targets in this table, initially thought to be essential for cancer cell survival, were actually not required for cell survival after all. They also discovered that a drug candidate in clinical development was effective at killing cancer cells, even after the supposed target protein was knocked out. The drug was causing cell death through off-target effects, and its proposed mechanism of action was invalidated. The study then identified multiple drugs with similar problems and uncovered the correct target for one of them, suggesting that more research and more stringent methods are needed to validate the targets of potential drugs before advancing them to the clinic. A case in point is the protein maternal embryonic leucine zipper kinase, or MELK. At one time, MELK was widely believed to have an important role in many cancers, and that belief was supported by numerous publications. They described a correlation between MELK expression and poor clinical prognosis, as well as the efficacy of MELK kinase inhibitors in cancer cell lines. One of these inhibitors advanced to clinical trials based on anti-tumor activities observed in preclinical models. All this was called into question when the same group from Cold Spring Harbor performed genetic validation, demonstrating that cancer cells remained sensitive to MELK inhibitors despite complete knockout of the target protein MELK, and that MELK is not required for cancer growth. Some of the confounding factors leading to target misidentification included problems with RNAi promiscuity and poor selectivity of tool compounds which were used to probe the target. Some of the common pitfalls in preclinical cancer target validation are well described by Nobel laureate William Kaelin, who highlights potential approaches to mitigate them. For example, researchers routinely use siRNA, or small molecules, to perturb the functions of specific targets in cells. A resulting change in phenotype is observed, which could serve as an assay readout. The effect is on target when there is true engagement with the intended target, and off target when the observed phenotype is due to engagement of an unintended target, which is usually also unidentified. It gets even more complex when a phenotype is induced by both on and off target engagement. A classic way to distinguish between on and off target inhibition by a drug is to demonstrate that the phenotype can be rescued or reversed. For example, when imatinib was developed as an inhibitor of Abl kinase activity, the rationale was that imatinib killed CML cells by inhibiting the pathogenic fusion protein BCR-ABL. But formal proof that the effect of imatinib was on target only came when BCR-ABL variants that were insensitive to imatinib displayed resistance to the drug. Hence, no cell death was observed. A stringent validation framework challenges researchers to validate or invalidate their scientific hypothesis at the start of the project. Selecting appropriate disease-relevant assays, for example, combined with careful interpretation of data, can build confidence that the correct mechanisms of action are being addressed. 48 new drugs were launched in 2019. In the year before, a record number of 59 new drugs were launched, providing more treatment options for patients. A third of new drugs were first-in-class therapies with mechanisms of action different from any existing ones. The reality remains that drugs currently on the market target less than 10% of the druggable genome. The effort must continue to develop new, innovative medicines to serve unmet needs in diverse therapeutic areas. Effective validation of potential drug targets will increase the chances of success. The Target Translation Consortium was established in 2019 as a national initiative to support preclinical target validation. Formed by 8 public institutions, the TTC works with researchers in Singapore to advance potential drug targets through effective target validation.